Thank you so much. Thank you. Thank you very much. Now, if we want to make the best products, we also have to invest in the best ideas. Every dollar we invested to map the human genome returned $140 to our economy. Every dollar. Today, our scientists are mapping the human brain to unlock the answers to Alzheimer's. They're developing drugs to regenerate damaged organs, devising new materials to make batteries 10 times more powerful. Now is not the time to gut these job-creating investments in science and innovation. Now is the time to reach a level of research and development not seen since the height of the space race. We need to make those investments.
And now I'm pleased to announce and introduce this evening's lecturer, Dr. Anshin Chang. Dr. Chang received his PhD and postdoctoral training at Rutgers University. He is a distinguished chair professor and the director of the Brain Research Center at the National Tsinghua University in Taiwan. He is also an adjunct international faculty member at the Kavli Institute for Brain and Mind at the University of California here in San Diego. Dr. Chan's lab investigates how genes and circuits orchestrate complex behaviors in Drosophila. He uses genetic tools, state-of-the-art imaging, and image reconstruction informatics to reconstruct a brain-wide wiring diagram with single-cell resolution. They then use this connectomics map together with powerful genetic tools to determine how the brain encodes and decodes information and how cell-type specific protein synthesis contributes to long-term memory formation. Dr. Chang has received many awards, including the Outstanding Research Award from the Taiwanese National Science Council and the National Chair Award from the Ministry of Education. Dr. Chang's talk tonight is entitled Towards a Whole Body Connectome in Drosophila. Please join me in welcoming Dr. Chang.
Thank you, Hollis, for the kind introduction. Life is a form of information flow. To understand information flows in the brain, we need to have a detailed connectivity map of neural circuits. As DNA structure to genomics, neurotyping is essential to connectomics. And the first one to classify various of different neurons is probably Cahoe using Gauge stem allow us to see diversity of many different kinds of neurons in all the many different kinds of the brains. And recently, many countries have these big brain projects in different countries, in different labs, use different model animals because our brain is so complicated to study. People use mouse brain because it's more close to human brains, use zero bloodfish because it's transparent, allow us to see the activity, and use Drosophila because it has all kinds of different sophisticated genetic toolbox, allow us to manipulate almost any gene in any neuron at any time now. And C. elegans remains still now the only animal with complete whole body connectons and with relatively simple neural systems. So each has its own characteristics. And the connectons has several different levels. It's kind of like a Google Earth, you know. And from macroscopic to nanoscopic, from region of the brains, track of the fibers to every single neurons to every single synapse. Different group of people, different institutes take different approaches. At the end, we actually need all different levels to understand the brain. This is the biggest challenge for neuroscientists to solve. So, to address these difficult challenges, we choose to use little Drosophila brain because all the brains share the common principle from gene to behavior, and which develop the new tools allow us one day be able to translate all the study in little brain to human brain and extract the basic operation principles from this genetic model animal. And our strategy initially is very simple. We are taking a reverse engineering strategy, trying to tear the brain apart into every single neuron and kind of put it back together to see if we can predict the information flow and use the map to understand how gene circuits to control behavior. And hopefully develop some technology not only allow us to understand the fly brain functions and be able to translate that to human brain study. So I started this about 20 years ago. Started by developing an aqueous solution called Focus Clear that render biological tissue transparent. Using a mouse brain as an example, this is a thick mouse brain slice at around 200 micron in thickness, and 96 stacks of confocal imaging stitched together. This is kind of like a Google Earth. One can zoom in and see very tiny fibers, almost every individual neurons. And because we couldn't find any objective lens with high resolutions allow us to see every single fiber like this with enough walking distance, we think making 200 micron in thickness tissues clear is enough, because beyond that, really, we don't have any objective lens to examine the structure. So with this technology, we start to map Drosophila brain circuits. And this is just 7 of these Drosophila brain neurons. And it actually took almost 10 years for the first confirmations by Asushi Miyawaki from RIKENS. And he first repeated our methods and confirmed it's working, and then come up with a replacement called scale, which use urea to cut the alpha bonds and kind of expand the tissues and make the whole mouse brain embryo transparent. And after that, many people come up with different methods. One of these most famous from Carl Deathrow called Clarity. He smartly embed the tissues in a hydrogel, and that's subject to more harsh treatment, extract all the lipid, and then put into the focus clear to make the entire mouse brain transparent. And nowadays, almost the entire mouse becomes transparent. With this series of tissue clearing methods, we can now ready to examine all the neural circuits in the animal brains. And about 20 years ago, luckily at the time, first GFP transgenic fly is become available, and Ziming Li and Lixin Lu developed a technology called MARC-M, which uses GAL80 to inhibit the GAL4 and to drive specific GFP expression in a subset of neurons. When randomly flip the GAL80, it allows us to label a few neurons at a time in the entire brain. It's kind of like a modern version of Gaugy stain. So combining GFP, MARC-M, and tissue clearing, we can then visualize individual single neurons at the entire brain. So taking this approach, we repeated this thousands of different times, and using different drivers that represent particular proteins involving in making a specific neurotransmitter. We flip one neuron at a time and repeated that. Initially, we imaged the 16,000 single neurons in the fruit fly brain and start to put them together and to see how the information, to predict how the information flow in the brain. This shows we use this as to identify 58 different brain regions and use these regions to recharacterize those single neurons. And we found that among these 16,000 neurons, about a quarter of those neurons belong to local neuron. Local neuron means both dendrite and axons are confined within a small brain region. And using this criteria, we can redefine the brain into around nearly 50 regions. And some of them with those local neurons we call local processing unit. Others without these local neurons, it has input and output from a specific neural pair we call hubs. And most recently, we are hoping to accelerate this process and developed an automated fly brain imaging methods by aligning flies like this and embedded it in a gel and cut a single vibratome slice and remove the head capsule. So these 25 flies lined up like soldiers in the Qin tomb now have the head capsules opened. And then the second cut allows us to get a fly head array. We can then put those fly head array under the microscope and pinpoint to the center of the brain. And so under the microscope, we can automatically scan the tissues 24 hours a day. And each brain we scanned it 4 times and then stitch them together into one intact brain. And this is what we call in situ brain imaging. The advantage of this in situ brain imaging is it can then align with functional imaging. With this automatic in situ brain imaging, we have so far imaged almost half of the total brain neurons in the fruit fly. That's more than 60,000 single neurons now. And with these 60,000 neurons, we start to do neuron typing, using the criteria that those neurons connect the same brain regions with morphology that we cannot distinguish it simply by eye as the same type. And those neurons connect the same regions, but we can easily differentiate that they are different in morphology as the same family but different types. And as different family. And if those neurons that connect different brain regions, we classify those neurons as different classes. And we use these definitions then to classify all the 60,000 neurons in the fruit fly brain. And it turned out we found around nearly 5,000 different classes with completely different morphology linking different brain regions. We then focus on a small region called protocerebral bridge, which is premotor structures within premotor center. And this little structure here, we map comprehensively almost every single neurons connecting to this little structure. As you can see here, and we label also the polarity of these neurons. It allows us to start to construct a very comprehensive map to predict the information flow. Okay, this the map is complicated, but actually it follows a specific rule. Okay, it's actually a repeatable rule. So with these repeatable and stereotypical patterns of circuits, we can then predict the information process within this little structure. But there are around nearly 50 more structures like this. So we are trying to take other strategies to accelerate this process. So we want to predict how the information flow in the brain using these structures, neurons as polarity that link to this protocerebral bridge as a ground truth, and to come up with an algorithm allow us to predict the polarity of all the single neurons that we have mapped, all the 60,000 single neurons in the brain we have mapped. And this prediction then allow us to predict the information flow in the brain. I'll give you one example. This is one of the visual circuit. And this particular neuron with dendrite, which is labeled green here, innervate the medulla layer sacs, where it has been reported previously that it received UV information. And this particular neuron with soma here, dendrite here, it gives the axon to this little region called OPTU. And we then search another neuron that has green with the shape like a red here, and we found this neuron. Okay, this is a cell body, the green is dendrite, red is neuron, and we continue the same search. And then you can find the entire circuit. And this circuit can be validated with grasp. And recently we developed a function, activity-dependent grasp, which split the GAP into half, half of the GAP in the vesicle, so activate the presynaptic neuron, it actually lights up with the postsynaptic neuron. And then we collaborate with physics, Szczytin and Olaf Spohn, let me see. Okay, it's difficult coming back and forth between Chinese and English. And my friend Ralph Greenspan, and we conclude that this flight brain neural network is similar to all the social networks. It follows this modular structures, rich club, loops, small wall theory, which is not surprising at all. So it kind of validated that it's a very useful system. The principle we extracted from this simple model system can be applied to other social networks, including us. So until now, I show you that we have almost half of the flight brain neural mapped and construct a database. We put all these neurons in the flight circuit database, and we release this database, so all the data in this database is completely freely available to anyone in the world. One can freely download, upload every single data, and upload its own data. We open an account for the people to upload its data, do all kinds of analysis, keep it privacy until he wants to release it. We also provide the data to many different places in the world for their own analysis. And we exercise this concept by collaborating with a few laboratories and come up with an open resource platform called Through Flight Brain Observatory in collaboration with Aria Laza at Columbia and Daniel Koka at University of Sheffield. We now have this observatory. One can pinpoint to almost any neuron of interest and then he can simulate where it linked to, and also immediately show the real data. And if you click the flight circuit here, you will see the real data in 3 dimensions. And my colleague, Luo Zhongquan, then used this neural network to simulate several different kinds of the brain disease now, including some spontaneous brain activity. So far I introduced you how we map the structures and put them together as an open source for everybody in different fields to use it. But the structures will be meaningless without functions. And to understand the function of this neuron, we need to be able to manipulate their gene expression and activity. So luckily, Fly has very sophisticated genetic toolbox. This GALVO US system is being widely used. So the GALVO can turn on various different effector genes through US system. And GALVO itself can be inhibited by GAL80. So if we identify a specific neurons expressing GALVO, we can then manipulate the gene expressions using optogenetic, thermogenetics, and many other different ways. So the key is, can we have such kind of tool? And luckily, Genelia Farm and Vienna people has generated thousands of different GALVO lines, and we remapped most of these GALVO lines with specific expression pattern. Okay, and we remapped about 5,000 such GALVO lines with quite specific expression pattern in the brain, and then using single neurons identified and cataloged in the flight circuit, and to search this GALVO driver line. So these 60,000 single neurons, each neuron has an ID page like this. This indicate all the connection of these single neurons connected to. So you can identify single neuron, you know where it connects to, and now for any neurons in the brain, you can easily identify a driver to manipulate that neuron. You can silence it, you can activate it, you can stop the neural transmission, you can inhibit any genes of interest. With those tools in hand, we start to identify specific functions. This just gives you one of these examples. To identify the information travels that when fly receives CO2 informations, in fly it has only one CO2 receptor, and we found that the different concentration of CO2 actually take different paths in the brain. Okay, at a low concentration, it turns on this path, projection neuron path 1. At a high concentration, it turns on 3 different paths, and turns out the third path is inhibitory. It releases GABA, inhibit the first path, so at a high concentration, information then take the second path here. So the entire pathway can be examined by specific GALVO drivers, carry optogenetic tools or thermogenetic tool to stop it or to accelerate the information processing and monitor the behavior outcome. Here's just another example about the memory study. So after we developed another new tool called ricin, which is ribosomal toxins, and it is core-sensitive mutants. We can turn the toxin on and off to inhibit the protein synthesis within a few hours immediately after training. When we do a large-scale brain screen by expressing these toxins in various different single neurons, and soon we'll identify several drivers. When we inhibit the protein synthesis, that animal will not be able to form long-term memory. And the overlap of these several different drivers come up with a single neuron called DALE, this pink neuron here. And this, again, is because of the previous database I described, allow us to identify this single neuron. We then come up with this neuron, we then identify the driver expressing only this neuron, and repeat the experiment again, showing that by blocking the new protein synthesis after spaced training, it abolished long-term memory. After this paper, we then come up with predictions, the following neurons or where the information coming from. We are now gradually to try to construct a comprehensive long-term memory circuit in fruit fly. So I don't have time to go through all the functional study, but use this map, we now identify a series of different functions in the fruit fly brain. It tells you how important this map is. It actually guides us to identify all kinds of different functions. So the rest of the talk, I want to share with you some of our thinking, that we think the brain is not only important in terms of communications between us and the environment. Perhaps it's more important that the brain needs to communicate with our body. So we propose that mapping a connectomic brain may not be sufficient. We actually need to map every neuron in the whole body. And here I want to show you how we do that. First, we started to build a machine, okay, in collaboration with a mechanical power engineering colleague of v Jianzhong. They help us to build a laser tracking systems so the fruit fly can walk freely in the arena. We teach these two fruit fly. Normally, the unmet unmet male will call vigorously virgin females, and they will copulate within 30 minutes also. But we teach this male fly to not do that. Whenever this male approaches the female within a body length, it will be punished by the laser. And he learns quickly. Okay, it's like a restraining orders from the court. And he formed long-term memory. In fact, after training, next day we ask him, he still remember. And the good thing is now you have 3 lasers, okay, and we can mimic the punishment by trigger the pen circuit. We first identify a mutant called penless mutants, which do not feel pain, and it will never learn. But with this pen galvo driver to drive general adoption, we can actually turn on the neuron without punishing the individual, but the fly will think he fears to be punished. And that way he can still learn to avoid the females. By doing that, we gradually delineate not just the sensory neuron, but the entire pen circuits up to the brain, and also the learning centers. There are a few genes in the learning center called mushroom body are necessary for this learning. So this kind of established this automatic optogenetic manipulation system. We have the capacity to illuminate only a small region of the insect. We could then use this to conduct a large-scale behavior screen. Inspired by recent papers from Barry Dixon's group, what they did is they screen various different galvo drivers and use that to express heat-sensitive effectors, and use thermal genetic. He can trigger specific neurons and elicit move forward behavior and backward behavior. So we validated that methods using the driver that he identified with optogenetics, and then apply this to all kinds of different behaviors. So far, we have identified more than 20 different kinds of behavior. Put it in short, that we can now remote control animal flight behavior. We can ask it not just move forward, backward, make a left turn, make a right turn, raise one wing, raise 2 wings, dance, shake the head, even lay an egg. I will show you a few examples. In this case, events, okay. It has just knocked it down. And this is move forward behavior. And this one is move backward behavior whenever it is triggered. And this one just spinning, okay. And this is jumping. This driver expressed in a neuron called giant fiber neuron. Whenever we trigger and activate this single neuron, it jumps. So, and this one is, I like this one very much because it always shake head. No, 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 okay. We triggered the arm, and by doing that, we illuminated the specific body parts with a laser that trigger specific neuron optogenetically. We identify a set of GAR4 line that express in different neurons that whenever it is triggered, it elicits a specific behavior. So we are now at the process of construct a neural behavior map that a specific subset of circuits that responsible for specific behavior. And how we do that? We conduct a neural behavior map. After behavior screening, if we identify a particular behavior, we then put the fly into an automated machine that allows us to map everything in the body. This is how we do it. My colleague, Lin Yanyin, at the Electrical Engineering Department, helped us to construct this fully automated imaging system. We put the flies in the clearing solutions, which embedded with gel, and then the machine can automatically imaging the fly and collect the imaging at the top layer of around 200 micron in thickness, and after that, it removes the top 100 micron layer and continue to the second layer. So the imaging part and the section part, it's always about 100 micron separated. And by the end of imaging, the fly is gone. And so we can then digitally reconstruct the entire fly. Now you can zoom in to see every neuron in this driver. Okay, I'll show you the next one. Okay, here just to show you 3 of such samples that imaging from top to the body. In fact, this is the same fly, 3 different labeling, okay? One is nerve system, the other is cuticle, this is muscle. And you can, like a Google Earth again, one can zoom in into the body, look at the individual fibers, and we also label all the muscles, so you know precisely the particular neurons innervate which muscle, and even to the leg. It depends on the driver, it tells you different, it labels different subsets of neuron. So if the behavior assay tells us already this is the neuron that elicit the spinning behavior, okay, or shake head behavior, then we can immediately identify all the candidate neurons. In fact, now we can flip individual neuron and repeat the same behavior, so identify precisely a single neuron involved in that particular behavior. So, until now, it sounds very promising, but it's also very depressing because it's so slow, okay? For the fruit fly brain, and we have worked on this for, I don't know, about 10 years also, and I calculated even for fruit fly brain, just to image the brain only, it may take 20 years. And then for the full body, it may take 40 years. And I don't have that much time. Okay. And we are all aiming at understand human brain. So is there a way I calculate this? If I do the same thing for human brain, it will take 17 million years. Okay, is there a way we can bypass all these troubles, come up with a totally different methods, okay? And this is what we want. I think this is the next generation brain imaging required. We need something with high resolution allow us to see every single neuron, every fibers at isotropic resolution. What that means is it's not just XYZ resolution, but actual resolution. When you turn to the 90 degree, we can still see it clearly, so we can isolate every individual neurons. And under such kind of a high magnification, high resolution objective lens, we want to see a large field of view. So far, we use 3D stitching. And it becomes even more difficult. We want to see unlimited depth. So we come up with this automatic imaging and sections, and basically allow us to observe any large tissue now. But this is not all. Image large tissue is not the only barrier. The most difficult part is we hopefully want to see how the neuron interact with the rest of the body. The best will be in its original place. And I think the most difficult part is speed. Can we do this really, really, really quickly? So far, with a little fruit fly brain, it takes us 10 years to be halfway. How can we do that with, on a human brain? Can we do this really fast? So I have these imaginations with all these challenges in head 10 years ago. And I was imagining if one day someone can do all this with a machine similar to MRI, which actually meet almost all the criteria except resolution, right? It's fast, it's large, it's in situ. It meets all the criteria. Can we come up with something like this medical imaging instrument? And then we approach my physics colleague come up with this solution. We use a synchrotron X-ray tomography. Synchrotron X-ray has less than 0.1. It's at angstrom levels of the wavelengths, so provide extremely high resolution. Okay, with a synchrotron that provide very, very bright light, actually penetrate almost anything. And my colleague kind of optimize the entire end station, and here just an example to show you this is entire mouse brain, labeled all the blood vessels, just to get you the idea of the size. We can now manage at least the size of the mouse brain. And this tells you the resolutions with every little bands here. The distance between bands is 0.5 micron, so we can resolve smaller than 0.5 micron. And for the entire fly head, and this is a TEM section just to show you the labeling is specific to a small subset of neuron. And this one is a Purkinje cells with the same preparation, just to tell you the resolution can be even higher than 0.15 micron to a nanometer scale. So with this tool in hand, we start to map the entire fly brain. Okay, so this is what I call micro CT. It shows in situ brain. You can see the brain is still within the head capsule. In fact, we can see the entire insect after we label the nerve system, and this is the muscle system and the intact cuticle. Okay, you can see also the nerve system linking to the body. Then you can zoom in to see how the brain looks like. And you see the entire brain. This method labels about 1% of neuron at a time. And the most beautiful part is the imaging takes only 10 minutes. That means within a day, we'll finish it, the whole thing. I'll give you a movie and show you how it looks like. This is just trying to show you the isotropic resolution of the neural fibers in the fruit fly brain. And remember, all the outside head capsules and neck and the body is still intact. We have digitally removed the rest of the other stuff, so you can see the fibers within the head capsule. And finally, I'll show you the last movie. So with this technology in hand, we can quickly image a lot of different fibers at the single cell resolutions. And this one just to show you when we align 4 brain neurons, we can align 40 brain neurons, but then you will not see anything, okay? Here just to show 4 of such neurons aligned together, and you can zoom in and see all the individual fibers. And so far we use manual segmentation. I think in the future it is possible to do this automatically. And this is just a partial neural tract segmentation from these sets of data. And some of these we can see the entire complete neurons. And for this segmentation, it can warp with in situ confocal image of the brain, the database I showed you previously, so you can compare the X-ray imaging data with fluorescence data to validate the segmentation. And it then allows us to see in situ nerve system throughout the entire tissue. And here just show you how we align different data, different type of data together. I will show you a final movie, and I hope you will like this movie. So remember, this is one single specimen, okay? You can see the surface, you can remove the surface, see everything inside. All these golden colors are muscles. We even identify new nerves. You can see this new nerve actually connected to the eye. And you zoom in into this brain, you see every single fibers has the same resolution from all angles. And you can segment the individual fibers outside from this data and align the several data from different brains together, or compare this data with optical imaging results. So we then put the neural peer models together with this X-ray imaging data to predict the information flow within the brain. So then you zoom out and you see the brain is still there. And in fact, we can now do this for any parts of the animal. I will stop here, and he will give you a brief summary. I show you automated in situ brain imaging, comprehensive cell typing throughout the entire brain, and we can now predict the information flow in the entire brain, and all the data are open and freely accessed to everyone in the world. And we have also developed an automated whole body mapping methods to relate specific neuron to specific behavior. And the last part is my favorite, So we developed an entirely new approach to image individual neuron in intact body at a large scale. Okay. I wish to thank all the collaborators, students, postdocs, and the funding support from the Ministry of Science and Technology and the Ministry of Education in Taiwan. Thank you very much.